Well, hello everybody, and welcome to Cheltenham Science Festival at Home. And this is unfortunately where we all are at the moment. We'd love to be seeing you in the green spaces of Cheltenham, but unfortunately this is where we are. But nevertheless, we are coming to you with all sorts of fabulous talks, and we're trying to bring as much as we can of the festival to you while you're at home. If you'd like to say thank you for the contribution of all these podcasts and things that we're doing, please do send us a donation, because it will allow us to continue the work that we do with Cheltenham and indeed with science more generally. So today we're going to be talking about DNA testing. None of you can fail to see all the tests that are now advertised on the telly, everything from ancestry to health tests. There's a lot that you've seen perhaps about the new genomic medicine service in the NHS. And there are also some concerns about what these DNA tests tell us, whether they're accurate, how we can be supported if we get an answer that we don't understand, and of course always questions about use of the data. So we are joined by two fabulous experts. I know them both of old, and you're in for a treat. My first guest is Sir Munir Pirmohamed. Munir is a specialist in a particular type of genetic test that's relating to pharmacogenomics. Now, if you've never heard that word before, it means really how your body, or how your genome dictates your response to medicines. And we've also got Joanne Hackett. Joanne is an expert in personalised medicine. That's the use of your DNA data in order to guide your choice of the best choice of treatments for you. And both of them are going to help us go through all these various aspects of DNA testing. So let's go to Munir first of all. Munir, tell me, just explain what these various tests are, what they involve.
So for DNA testing, you can have very many different types of tests. You can test for each individual variant in your genome. You can go and do a scan for the whole genome, where you can look at 1 million different variants in your genome, or you can do sequencing of your whole genome. And the complexity increases as you go from a single variant right through to the whole genome, and the cost obviously also increases as well. The amount of information you get also increases from doing a one variant right through to the whole genome as well.
So what we've seen typically in hospitals is people go in for genetic tests because they have genetic disease, but that whole thing is changing now, isn't it?
Very much so, very much so. It has been very much based on individual genes and looking at a particular disease. But as we move forward, it's moved to panel testing. You have multiple genes being tested when you go in for a particular condition, and much more. What's going to happen now, and the UK is the leader in this with the Genomic Medicine Service, is that some patients will be able to get whole human genome sequencing being done within the NHS.
So we've got everything being tested, from just one thing to basically the whole lot. And those tests have also got much cheaper, haven't they? Because it used to be that they were really complicated, took a very long time, and now that's very different.
Very much so. So, it used to be that even doing a single variant in a gene took a day or so, but now we can actually do a whole human genome sequence. The initial cost when the first human genome sequence was done was about $3 billion. It took about 10 years or more to be able to complete that. We can now do a whole human genome sequence for less than £1,000, and it can be done in less than a week.
The tests that we have, they're much more common, and they're now being offered over the counter. But before I just leave the tests that are offered in hospital, I want to turn to Joanne. Joanne, tell me about the kind of tests that are used in hospital for cancer now.
Great question, Viv. So a lot of people are trying to understand the differences between looking at particular point mutations or looking at various genes that are known to be associated with different aspects of cancer. So whole genome sequencing, as Munir pointed out, is something that looks at everything. However, with cancer, it's a little bit different. Sometimes the everything is not actually necessary to be able to look very specifically at certain types of cancers. And also the way that we look at these mutations is very different in cancer. You need to look very deeply into the genome. And that may sound a bit silly to say, what does it mean to look any deeper? We're looking inside of our bodies anyway. But really what it means is you have to do, if you think about photocopying, it's doing many, many layers of the photocopying effectively to be able to see down into the different variations into the genome. And you need to do that with cancer genomes. So finding the different genes that are useful and or the ones that are affected in order to have cancer tumors are the way that you have to look at that, and you have to look at it very deeply. So very specific gene panels, which is a variety of different genes coming together and being sequenced very deeply is the method that is happening quite a lot for cancer genomics today.
So what we should explain perhaps here is that cancer is a disease of an altered DNA. So you get one change and then you get more changes in your genome as the cancer takes over the machinery of your cell to increase and prosper. And what we're looking for when we're looking for, we're doing DNA testing with cancer, we're looking for different changes, because those changes are going to guide what medicines will work best for that particular cancer. And we already use some medicines that are specific. So for instance, Herceptin is a very well-known one for breast cancer, but this is now coming to many, many more cancers, isn't it?
Yes, absolutely. And the other thing with cancer is we have to look at the normal aspects of the body as well. So the cancer tumor is usually the only thing that has mutations in it, aside from the fact that we all have some normal mutations in our DNA anyway. So we have to take a sample that is considered normal, and then we compare the two of them. But what's quite sneaky with cancer is that very often it's not just something missing or a duplication of something. Sometimes there's aspects of the DNA that fuses to itself. So these are called fusions. And you can only see that a piece of the genome has fused to another piece if you do this really deep sequencing. So you look at that, you look at the normal, which is usually taken from a person's blood, and you make the differences between the two, and that's how you're able to determine the genetic form of cancer that the individual has.
And the Genomic Medicine Services has been rolled out in the NHS is going to allow that kind of cancer assessment to be much more normal and to be offered to everyone who needs it.
Absolutely, yes.
Okay, so those are the hospital-based tests. Those are what's in the medical field. Munir, we see tests being offered all over the place for lifestyle things, for health, 23andMe, all those kind of things. Ancestry, what are those tests?
So those are called direct-to-consumer tests. You can buy them on the internet, and you can send off a mouth swab to the company, and they'll send you results back. With the important thing to note is what you'll get, the information you'll get will be completely different between different companies. The coverage is also different between different companies, and the information and the advice you get is also different between different companies. So there is a note of caution there in terms of being able to interpret what you get back from the companies.
These tests, so is it buyer beware, and how do we assess whether a test is any good?
So they largely been brought in for checking your ancestry, and that's what most people use it for. But increasingly, a large number of these companies are providing tests which may have medical consequences. For example, some may be able to test for your BRCA gene, which increases the risk of breast cancer, ovarian cancer, and so on. There are other tests which are also starting to include variants which determine how you respond to particular drugs. But the coverage between those different tests is completely different. Some have a large number of variants, some have a small number of variants. And sometimes the evidence base for those variants is not good enough for us to be able to include them within our clinical testing that we undertake. So there is a note of caution in terms of what you get from these particular companies and how you interpret those particular tests. So
when you say coverage, what you mean is how many things they look for.
That's right. That's right. So, and there've been several studies which have been undertaken comparing tests from between different companies, and the number of genes being looked for in those different tests varies, between different companies. So, and sometimes when you have many genes, sometimes the evidence base may not be very good to be able to include those genes. But even when there is good evidence base when some genes are included in other tests, the information you get in terms of advice you get based on those can vary as well between companies and so on. And so it is important to be aware of those limitations.
And actually, Joe, knowing what they're looking for is really important, isn't it? Because when people say they are looking for BRCA genes, actually, there are many, many, many BRCA genes. And
yes,
if someone is only looking for, say, 3, then actually, when the result comes back that says, we're pleased to tell you you haven't got these particular BRCA genes, you think, oh, that means that I'm not going to get breast cancer. But that's not what it means at all, is it?
correct. So there are different risk scores that can be assigned to particular genes as to whether or not you have a higher probability of being able to get a particular type of cancer based on the way that your body is made up and the way that other things interact with it. And by not testing everything, you can actually get some false positives or false negatives. But that's also not to say that you need a whole genome in order to get accurate results. But this is why, if it's going to have clinical relevance, it has to go through a hospital or it has to go through a clinical setting in order to be able to verify that the actual set of genes that they're looking at are verified and scientifically valid.
And another problem, Munir, is that it depends, again, what you're looking for, doesn't it? Because there are some genes that are perhaps found, I mean, BRCA is a good example. So there are some genes that are found very commonly in people of, I think it's a Jewish, is it Sephardic background. And actually, if you're testing for those in a population that isn't from that background, then it's really not that much use.
No, and that's absolutely correct, because we know that the kind of genes or the variation in genes varies across the world according to different ethnic groups. And so testing in one ethnic group, you may find a high frequency of that particular variant, but testing in another ethnic group, you may not find any of those variants at all. So it is very important in terms of understanding what the coverage is, what the particular variants are covered, and where it will be relevant for particular ethnic groups.
But also on what population they based their information. Because if you're comparing, and many, this is an issue right across genetic testing, that many of the libraries, if you like, that have been the basis for the development of these tests are actually white Caucasians. Quite a lot of them come from Cambridge, quite a lot of them, both in Cambridge, England, and Cambridge in Massachusetts. And actually, they may not be relevant to the population that is actually using them at all.
Yeah, no, absolutely, that's correct. So if you look at all the genomic information that's now available, often on online databases, free access, you find that the majority of the information available is in white populations. There is increasing amounts of information being available in other populations, but there are many populations throughout the world which are underserved with regard to genomic information. So you really have to be careful with regard to that.
And what's normal for one population can be not normal for another population. I remember there was a particular change that's associated with epilepsy in the UK, but in Indian populations, it's just normal.
That's right. Yeah, very much so. And that has been seen in many different areas, many different disease areas, but also in pharmacogenomics as well. For example, there's a gene in the Southeast Asian population, particularly Han Chinese, which predisposes to a severe adverse drug reaction with the drug called carbamazepine. It's seen in the Southeast Asian populations, but if you then start looking at the white populations, the frequency of that is so low that it's almost non-existent.
Gosh. So Joe, just explain to me that, all this genetic testing has now come up, and it's partly because we can do it now because we've got so much better at doing it. But what's the purpose of all this genetic testing? What's it for?
Yes, so this is the area that I always get very excited about, because I think we have accumulated a vast amount of knowledge, which I believe we had to before some of the really exciting developments are going to happen. And in order to really create some of these exciting opportunities, this colossal data set needs to be amassed. And as you've rightfully pointed out, it has to come from a variety of different backgrounds. It has to come from people who we think are normal and potentially are holding mutations that we've never seen before, or they have to come from people who we know are sick to be able to validate that. And once a lot of information has been accumulated, and we currently have globally probably quite a lot of data now, it's still not enough, there's never enough data really. This is now when there's an opportunity to really take a personalized approach to it. And I don't mean personalized in the sense that you get to know every single thing about me and therefore I get all these wonderful treatments specific for me. It means you get to actually look up against a population and also start to stratify, which is like really trying to identify patients that look the same or react the same to particular medications or have different symptoms, and you can actually potentially diagnose them earlier or faster. And all of this data is necessary to give you, as an individual, the best possible treatment without necessarily having to completely make a treatment that is unique just to yourself. So a lot of the pharma companies, we hear people talking about the fact that, oh, these drugs don't work, or I don't understand why this clinical trial failed. Well, do we not have a responsibility to be able to give these individuals as much information as possible to make the drugs as useful as possible for the individual that would require them? Of course, the answer is yes. So this data is now allowing us to garner those insights to be able to treat the right populations with the right medication, potentially have different dosages for different individuals, going back to what Munir said earlier, and also just being able to understand whether or not there are other factors that are part of this. So if you drink a glass of wine and have your medication and you get a rash, is that because of the medication? Is it because of the wine? Or is it because of the fact that you're dehydrated? No one really knows any of this stuff until all of that data gets collected. And I think that we're finally at that tipping point that we're starting to see some real examples of insights that have been garnered based on the fact that this data has been accumulated and allows both researchers and industry professionals to be able to access this in a way that they can actually look at volumes of data to help them make those decisions.
So it's a fascinating area. I mean, this is the beginning of personalised medicine, isn't it? But as you say, not personalised so that everybody has an individual, pill, but personalised. For example, we know that diabetes now is not just one disease, but a number of diseases. And, it's your genetic background that really puts you in one particular, if I can describe it so unglamorously, bucket of disease, one bucket rather than another bucket. There are lots of different diseases that, as you say, we treat all the same at the moment, but actually this will allow us to treat groups of people in a different way. And is that going to be just using existing drugs, or will we see new drugs for these different groups?
Well, what's really been quite exciting is that there have been a lot of drugs that have been created that have quote unquote failed. And when they fail, people effectively just take them, put them on the bookshelf and don't really think of them ever again because they've been deemed to not be a success at all. Well, all of a sudden there's enough data now that if you actually look at that data independent to actually doing a real clinical trial with real people, the same results can actually be achieved. So if all of a sudden you look at the compound that was made 10 years ago that you thought was going to be effective for contact dermatitis, for example, if you all of a sudden realize that a lot of the individuals who have epilepsy, for example, have a particular mutation, and that mutation just happens to be the target for that drug, if you start giving those individuals that drug, they may all of a sudden respond. But no one would have thought that the drug that was originally developed for one thing could be used for something else. And we're starting to see more of this, which is fantastic because the cost of a drug is extremely expensive, as we all know. And if we can actually find the individuals who will respond to that medication faster and put them on it at the beginning, not only is the drug development process going to be cheaper, it will be more effective, and also the individuals receiving it will actually get better, which is what we all want. We don't want someone to take medication and have bad side effects or to have no response at all and have to go through another cycle about worrying about, let's try something different. Is there going to be new side effects? Is it interacting with something else? So it's a smarter way of doing it. So if we're all of a sudden seeing these repurposing of compounds, we're hoping that this will actually allow the trials to be cheaper and the drug development process as a whole to be much more streamlined. But in addition to that, if you know the exact target and you know the gene that you're actually trying to work towards as your target, you're going to be able to make your drugs and your compounds in a much more effective way. And again, this hopefully will reduce the cost burden and the number of individuals required for clinical trials. And I'm actually quite excited about the concept of the virtual clinical trial, which is already starting to happen. Again, using data, knowing that individuals all have a particular mutation, and then being able to treat them that way. So if we can start using virtual control arms to do clinical trials, again, this will help reduce the cost and help reduce the amount of time it takes to bring these new compounds to the market, which is really exciting. And this is why this colossal data set that we, as in folks in the UK and also folks around the world, have been aggregating and trying to put together in a federated way so that all of this data can be sat and looked at to be able to make these insights.
What does federated mean?
Federated is effectively, there's a couple of ways of federating data, and I think this is really the promise of genomics, is that all the data around the world can come together in one big pot and you can look at that data. But the beautiful thing is it doesn't actually have to be identifiable. So you don't have to know it's Joanne, who is a woman and who is a certain height and a certain weight and has brown eyes. You don't need to know any of that. You just need to know that I have a mutation in a particular chromosome. And once you start to see more and more and more people with that, you can actually make some analysis. So federation is basically putting all of the data together. Think of it as in one big Excel spreadsheet, really, but cutting off anything that's identifiable. So it's the way, I think, to really combine both clinical and genomic data to make some really exciting insights. And that's definitely the trajectory for the world of clinical genomics as we see it.
Thank you. I want to talk about data a bit more in a moment. The whole pharmacogenomics thing is something that's kind of been around at the edges and it's not really taken off. But are we going to see DNA testing for pharmacogenomics to find out what each of us, what medicines will work best for us? Are we going to see that coming to the NHS soon?
Yes, absolutely. I think this has been increasing over the years, and many parts of the world already have that available. Certainly within the NHS, NHS England genomics laboratory hubs have been looking at the possibility of introducing pharmacogenomic testing for our whole population when they're taking certain drugs where there is good evidence that pharmacogenomic variation affects either the efficacy or safety of that drug.
So give us an example of a type of drug that might be like that.
So an example for safety is something like abacavir, which is a drug used in HIV. It causes a serious immune reaction in about 7% of individuals, characterized by skin rash, respiratory manifestations, tummy upset. But we have identified a gene which is on chromosome 6, and if you type individuals for that gene and exclude individuals who are positive for that gene from getting that particular drug, abacavir, you can drop that rate of hypersensitivity of the immune reaction from 7% to almost 0. So basically, that has made that drug virtually, completely safe because of that genetic test. For efficacy, there's the issue of codeine. Codeine is a very widely used drug for analgesia. About 8% of the population in the UK will not respond in terms of pain relief from codeine. And no matter how high the dose is, they just will not get any pain relief at all. That's largely because they lack an enzyme which breaks down codeine into another compound, which is what determines whether a codeine works or not. So basically, codeine is something called a prodrug, and the enzyme breaks it down into its active drug. And 8% of the population in the UK actually do not have that particular gene. So you may give them large amounts of codeine and they'll never get any pain relief.
And so there are lots of us who are saying to people, you're such a wimp, you've had all that codeine and you're still moaning. And actually, it's not that they're a wimp at all. It's just that they may be one of those fast metabolizers of codeine, and it's not touching the sides, as it were, and they are still getting a lot of pain. And that actually, might that also have an implication for things like, fentanyl? We've heard of these use of opioids, but actually, if we knew who reacted, who needed smaller doses and larger doses, might that help in that particular situation?
Yeah, so the codeine enzyme aspect I mentioned is specific to codeine. With fentanyl and with the other opiate drugs, there may be other polymorphism in the receptor, in other enzymes which may be important. The evidence base is not so good for that, but there's a lot of work going on. And obviously, we are facing an opioid epidemic at the moment. And I know there's a lot of work going on all around the world to identify whether there are particular individuals who are susceptible to developing opioid addiction. And what kind of doses they need. And another thing that genetics determines is what kind of dose you need for relief of your pain, or when you're using a drug such as warfarin, which thins your blood and so on. So the genetics can have an effect on dose, on the efficacy of the drug, as well as the safety of the drug.
Well, you've answered absolutely a question that we had from Rahima Begum. So Rahima, if you're listening, thank you for submitting that question. And she asked, can the analysis of genetics in personalised medicine prevent or reduce adverse effects to certain medicines? And I hope you now know from Munir that the answer is a big fat yes.
Yes, absolutely.
So there's also a question from Esme Benwell. And this is one for you, Munir. Is there any genetic basis to the interactions of foods like grapefruit with anaesthetics?
So grapefruit is a very specific example. When you take grapefruit, it inhibits an enzyme in your gut that normally breaks down certain drugs, such as some anaesthetics, such as some of the sedatives which are used in anaesthetic, for example, a sedative called midazolam. But I don't think it matters with regard to your genetics. If you do take a grapefruit juice, it will inhibit that enzyme, and you will get much more of a sedative effect for some of the anesthetics which are used. So often the advice is avoid grapefruit juice for at least a few days before you go and have an anesthetic.
Interesting. So we've got another good question from Leah Fowler Roberts on a very similar theme. My mum had an operation and needed more anaesthetic to put her under, and she was told it's because she's a redhead. Does red hair really coincide with an increased resistance to painkillers and anaesthetics?
Okay, yeah.
This is a natural redhead, we're assuming, Leah.
Yeah. So the natural redhead we know what the genetic basis of that is. It's a mutation in a gene called melanocortin receptor 1. And studies which have been done, they were done, maybe about 10, 15 years ago, suggested that people with red hair are more difficult to anesthetize. They require larger amounts of anesthetic, sometimes even local anesthetics that require large amounts of local anesthetic. But there may be, in terms of pain, there may be it depends what the kind of type of pain is. For certain pains, they may actually be resistant, while for other types of pain, they're more sensitive. And the biological basis of that we still don't understand. It seems that the melanocortin receptor is also important for interacting with the body's endogenous hormones or molecules called endorphins. And that may be the basis, the biological basis of that differential sensitivity to anesthetics as well as pain relief.
Absolutely fascinating. I am a natural brunette, not a redhead, but that's really interesting information. So let me turn now back to these particularly lifestyle and ancestry and all those kind of things, because a lot of people wonder what is happening to all the data. Joanne, should we be concerned about what's happening to our data when we sign up for some of these seemingly innocuous lifestyle testing kits?
I would always say to be a bit cautious when it comes to signing up to anything that involves personal data, because there are a lot of things that people just don't know. It's very rare that people are trying to be sneaky, but there's a lot of things that we just haven't really considered very often before making these decisions. So, for example, if you read a disclaimer that says we would like to be able to aggregate or federate your data. And you think, that's fine, that'll never be attributed back to me. I'm happy with that. I'd like to be one of many to be able to contribute to something. So you may tick that box. But if there's only one box to tick, and a little bit later on in this disclaimer, it says, we will also be able to recontact you because we actually know who you are. Well, if you didn't really read that, or you weren't entirely sure what that meant, and the two of them maybe were contradictory, you may have ticked that one box at the bottom and actually agreed to two completely different things. And it's actually sometimes quite challenging for people to really understand what's being asked of them. And I think there are some very clear examples as to why and how this can be done, and it is very transparent. Individuals need to have the complete right of their data. They also need to know that there is a trade-off. So if you want someone to know more about you, you do have to disclose personal information. But the way that's being treated and the way it's being used, it can and should be done in full transparency, and it should be done in a way that individuals that are permitted to look at specific aspects of that have the right to do that. So it is a little bit of a challenge, because there's sometimes these disclaimers are 20 pages long, and it would take, a very educated lawyer to probably sit there and go through line by line. But I think fundamentally we need to ask ourselves, if this is a lifestyle test and we're looking at getting information for ourselves, and it's maybe a little bit for fun, or maybe it's just to ask a simple question, you need to ask yourself, is that trade-off worth it? Is me getting that information worth me trading off all that information that about me that has been collected? And if that doesn't feel right and it's not comfortable, it's something you shouldn't be doing. A healthcare system is very different, obviously, but again, you have to make sure that the data that is being collected is going to be used for the right purposes that you're comfortable with. And I think this is why there are now more tick boxes, I think, at the bottom of all of these disclaimers, and you have the right now to choose a little bit more wisely as to whether or not you're comfortable with one particular option or the other. And I think the more that happens, and the more a patient feels empowered to make those decisions, the more comfortable individuals will be to start trading that information for either a result or for a bit of fun or for whatever it is. And I think that we haven't quite got to that point yet, I don't think.
Because Munir, should we be worried about the way that some of these, particularly some of these very cheap ancestry tests, that they're sure, we're being told we're related to Vikings. Spoiler alert, everyone, we're all related to Vikings. But might be told we're related to Vikings, or if you're Danny Dyer, to every crowned head in Europe. But those are very cheap. But actually, those people can sell off that DNA data to other companies, and they're not regulated, those lifestyle things, are they?
No, so yes, I think I agree with what Joanne said, that you really have to be aware of what you're signing up for, read the small print, and make sure you know what your data is going to be used for, and what it's going to be, how it's going to be forwarded. And as you mentioned, there are companies which do now sell off the data for further research, for development of new drugs and so on. And so it is important that you are aware at the beginning, when you sign up, how your data is going to be used. And once you have signed up, etc., you also need to have the option to come out of that particular data bank as well.
And we're actually, I think, going, I don't know where we'll be in terms of the Brexit regulation, although I suspect this will still apply. But certainly there is a new European directive which is going to treat medical information, so not the lifestyle stuff, but medical information from genetic testing kits. The genetic testing kits are going to be regulated to a much higher standard, I think, from 2021. And I think that's to be welcomed, isn't it?
Yes, so this is the in vitro diagnostics directive that you're mentioning. Yes, absolutely. That is important, to have that good quality of test that gives you the right answer. But also, but there does need to be a degree of proportionate regulation as well. What we don't want to do is to over-regulate, because if you over-regulate, you will reduce innovation. So there is a balance to be struck where you need a proportionate degree of regulation, so you don't stifle innovation, so that we make these advances, at a speedy rate, to be able to help human health, really.
So we're coming now towards the end of our session, but before we go, I wanted to ask a question from Molly Allwood. Molly, I hope you're watching this now. Will the new DNA testing ever be able to pick up and prevent genetic diseases before they begin to affect a person? For example, osteoporosis or breast cancer. And Jo, would you like to take that? Because the 100,000 Genomes Project had an additional findings element to it. And the additional findings was not about diseases that the person actually had. It was about looking at things that they might have that the NHS could do something about. Not necessarily prevent completely, but perhaps put in additional screening.
Yes, so I think, to be very clear, no amount of genetic testing will ever prevent an individual from having a genetic disease. That's unfortunately, you pick up the genetic defect or mutation because it's already there. So testing won't help prevent that. But what it may do, and you very rightly pointed that out, it may actually help treat that before it becomes symptomatic. So there are individuals that have metabolic diseases and they go for a very long time without being diagnosed. And sometimes something very simple, not medication, a very simple lifestyle change, potentially a ketogenic diet, or something like that is all they need.
Ketogenic diet. What's a ketogenic diet?
Oh, so let's not get too involved into various different methods of dieting, but it's different forms of diets, of those that restrict carbohydrates or restrict fats or have more fats or more carbohydrates. There's different types that actually respond very well to different metabolic diseases. So a ketogenic diet very often is what is needed. Sometimes the opposite of a ketogenic diet is what is needed. But if you know that, it's something very simple. It's not taking any different supplements. It's not doing anything that is very strange at all. It's just eating or restricting certain foods that your body's not going to process. And if you know that, you would do that, and you would probably do that from birth, because that would actually be a much smarter thing to do than to suffer the consequences of having tummy aches or headaches or various things that happen because of that. And if you know that in advance, you can actually prevent sometimes the symptoms and or the consequences of having a disease if you catch it early. In the same way, I think there's a lot of different cardiovascular diseases that there are no symptoms, and all of a sudden, by the time you're 40 years old, you're very high risk of having a heart attack. But if you knew that in advance, and the clinical systems that you're working with and through know that, you would be monitored. And it's much better to monitor an individual and to make sure that they're as healthy as possible rather than getting them sick and trying to treat them. So genetics holds the opportunity to really be able to push the way that we treat individuals differently so that we're not treating sick people, but we're preventing them from getting sick. And the whole concept of obviously preventing people from getting sick means they live happier and healthier lives, longer lives, and the healthcare system is actually there to treat sick people as opposed to try to put plasters on individuals. So I think we're working towards a way that we'll be able to identify things sooner and be able to prevent people from getting really, really sick and having to go through that horrible situation of trying one drug. Maybe they have a genetic mutation that shows they shouldn't have the codeine, but we don't know that. So rather than just having this really quite difficult way of treating an individual, let's just not allow them to get sick to begin with.
Munir, a lot of people are very frightened by genetic testing because they think that something is going to be revealed that is a dread disease that they would really rather not know. And that's, it holds people back from having genetic testing. But of course, it's not that when people are tested for something like, let's say for pharmacogenomics, nobody's rifling through your genome saying, aha, we know secretly that you're going to, have this dread disease. So people who are looking at the information do not have information that you don't have.
Yeah, so I think that's a particular issue when you do whole human genome sequencing, because you may find additional variants which you may not be aware of, or you may lead you to sort of potentially develop disease 20 or 30 years after you've had that particular test. People have done surveys of what do you do with these incidental findings, as they're called, and surprisingly perhaps. A lot of the people who've had whole genome sequencing say that they want to know about these incidental findings, because at the moment we may not be able to treat some of those incidental findings, but actually medicine is advancing rapidly, and I think it is worthwhile knowing about those incidental findings, because there may be lifestyle measures you can undertake to be able to reduce your risk, for example, of metabolic disease, based on the fact that you have a particular
Joanne was talking about metabolic disease. Just explain to people who might not know what metabolic disease means. What are we talking about here? Like diabetes and things.
Yeah, so metabolic disease is something which is very much associated with being obese. And diabetes is one aspect of that, but there may be many people who are not diabetic but still have something called metabolic syndrome, where you have higher levels of sugars, which are not quite in the diabetic range, higher level of cholesterol, higher levels of high blood pressure, and so on. So all that increases your risk of developing heart attacks, strokes, and things like that. So I think there may be particular genes which predispose you to those kind of complex diseases. But if you know that you do have a predisposition to that, you may be able to do something about it now by changing your lifestyle, improving your diet, increasing your exercise, and so on. And the important thing from all of this is the genes are not everything. Actually, what you do in your daily the way you live is also very important to be able to protect your health for the future.
Yeah, that's a point we can't stress enough, that your genes are not your whole story. So,
that's right.
Just because you have a predisposition to something doesn't mean that you will get it. Now, yeah. I want to finish up by asking, we have to ask about COVID-19 and genes. Is there any relationship between people who get it severely and their genetic background?
Do you want me to answer that first?
Yes.
Yeah, okay. So there's an awful lot of work going on with regard to COVID-19. As most people will know, the majority of people are very mildly affected. It seems that 20% of people become hospitalized, and a smaller proportion of those then end up on ITU, and then unfortunately some people have died from COVID-19. If you look at the risk factors, the majority of risk factors seem to be associated with age. Men are more predisposed to develop more severe disease, obesity predisposes to severe disease, and having many different diseases together. For example, if you've got underlying cardiovascular disease, hypertension, diabetes, that increases the risk of developing much more severe COVID disease. However, there are some young people who have got very severe disease from COVID and unfortunately have died, suggesting there are other factors. They haven't had any underlying health problems. So genetics is certainly an important part of that. And there are many sort of, studies going on in the world to identify genetic predisposing factors. And this week, Genomics England announced a collaboration with the particular study that's going on, where they are going to whole human genome sequence individuals with who've had COVID disease of different severities to identify what the genetic factors are which predispose to you to develop a severe form of COVID-19. Also important in that will be the incidental findings. For example, there are drugs which are being trialed at the moment with regard to COVID-19. Hopefully, we will have some drugs which are effective in treating COVID-19, but there will be people who will vary in the responses to those drugs, and the information that's going to be gained from the whole human genome sequencing in terms of pharmacogenomics will also help in terms of trying to identify which drug you're going to respond to best when we have more than one drug available. I'm very optimistic that we will have more than one drug available to be able to treat COVID-19.
Terrific, thank you very much. So I think what we can say in conclusion, that genetic testing is here to stay, that it's moved way beyond the genetics department. It's no longer something that's just confined to looking for diseases that are inherited. It's much more about looking for risk and for preventing disease, as Joe said, and trying to make the best use of and best treatments for people based on their genetic inheritance. So thank you to both of you, to Munir Pirmohamed and to Joanne Hackett. Thank you so much for staying with us, and enjoy the rest of the smashing programs that are available to you through the Cheltenham Science Festival at home. Bye for now.
Thank you.
Thank you.